Let's take a minute to look at the unique structural characteristics of DNA. DNA, unlike RNA, is double-stranded. There are two strands of nucleic acids lined up in opposite directions. So one side of the sugar phosphate backbone will be going in the 5' to 3' direction, while the other side goes from the 5' to 3' direction. What is holding the double helix together is hydrogen bonding, and hydrogen bonding is typically represented by these dots or dashed lines. And we see that there are two base pairs that will always hydrogen bond between each other. It will always be a purine-based hydrogen bonding with a pyrimidine base. So we see adenine forms 2 hydrogen bonds with thymine, and guanine forms 3 hydrogen bonds with cytosine. So these complementary base pairs will always line up and hydrogen bond to keep the double helix in position. The chirality of the sugars on this backbone is what causes this twisting of the DNA strands, and that's known as a double helix. And if we were to abbreviate a specific section of DNA, we would specify which direction we were going from. So we would call this right here 5' A, C, T, G, T, etc. And this side would be 3' T, G, A, C, A, etc. And if we're given one side, we can always figure out what the sequence is on the other side because of the complementary base pairs. A will always pair with T, and C will always pair with G. So looking at more detail at the hydrogen bonding between the base pairs in adenine and in guanine, the two purines, the hydrogen bonding is going to occur in the 6-member ring. And in adenine, we see that's going to be on the 1 nitrogen as well as the amino group that is on the 6 carbon. In the thymine, the hydrogen bonding with the adenine is occurring from the hydrogen on the 3 nitrogen as well as the carbonyl group on the 4 carbon. In guanine, the hydrogen bonding is going to occur between the carbonyl on the 6 carbon, this nitrogen in the 1 position, and the amino group on the 2 carbon. In the cytosine, the hydrogen bonding is occurring between the carbonyl on the 2 carbon, the nitrogen, 1, 2, 3, in the 3 position, and the amino group that is on the 4